Finally, we move on to specification number 5, which asks us to describe the use of enzymes in biological washing powders, pectinase for fruit juice production, and lactase for lactose-free milk. To begin with, enzymes are biological catalysts, meaning they speed up chemical reactions in living organisms. They are highly specific, so each enzyme works on a particular substrate, and they can function under mild conditions such as low temperature and neutral pH. Because of these properties, enzymes are widely used in biotechnology, including in medicine where they help in processes like producing insulin and antibiotics. One important use of enzymes is in biological washing powders. Biological washing powders contain specific enzymes produced by bacteria that can break down large, insoluble stain molecules into small, soluble ones that wash away easily in water. Proteases are enzymes that target protein-based stains such as blood, grass, egg, and sweat. They break down long protein chains into smaller, soluble amino acids. Lipases are enzymes that target fat and oil-based stains such as grease, butter, or oil. They break down lipids into fatty acids and glycerol. As you can see here in the diagram, there is a before and after comparison of a fabric fiber with a stain attached to it. Enzyme molecules approach the stain and break it into much smaller pieces. In the after stage, the fabric appears clean because the stain has been broken down and washed away in water. Next we will discuss pectinase for fruit juice production. When fruit is crushed to make juice, a large amount of liquid remains trapped inside plant cells. In addition, pectin present in the cell walls acts like a glue that makes the juice thick and cloudy. Pectinase is added to the crushed fruit, where it breaks down the pectin in the cell walls. This allows more juice to be released from the cells, increasing the overall yield. It also makes the juice clearer because the substances causing cloudiness are broken down. As you can see here in the diagram, a plant cell with a thick cell wall is shown being broken down by pectinase enzymes. The diagram then shows the change from cloudy juice containing suspended particles to clear juice after the enzyme has acted. Now talking about lactase for lactose-free milk. Lactose is the natural sugar found in milk, but some people are lactose intolerant because they do not produce enough lactase enzyme in the digestive system. This can cause digestive discomfort after consuming milk. To produce lactose-free milk, lactase is added to break down lactose into two simpler sugars, glucose and galactose. This makes the milk easier to digest for lactose-intolerant individuals. In industrial production, lactase is often immobilized by fixing it onto beads. Milk is passed through a column containing these beads so that the enzyme breaks down the lactose without mixing into the final product, and the enzyme can be reused. As you can see here in the diagram, a lactose molecule is shown being split by lactase into glucose and galactose. In the industrial setup, milk enters a column containing enzyme beads and comes out as lactose-free milk after passing through. So this was all about Unit 18.1, Biotechnology. Stay tuned for the next unit. Thank you for watching.